Did you know that soils are the basis of all of the food that you eat? They also provide a range of other services such as purifying water, storing carbon from our atmosphere and providing a home for biodiversity. They also support all of the buildings and homes that we have on the planet. Here at Teagasc Johnstown Castle, we've been working on soil science for the past 50 years. The initial focus was how to improve our soils to increase food production after World War II. Our focus has now changed to more sustainable food production systems. In our centre, we have a range of scientists working on different themes such as biodiversity, carbon sequestration and greenhouse gas emissions, water quality, and on soil science itself. Our research here focuses on soil health and its importance in both underpinning sustainability and producing food. In this video, our scientists will give you an overview of the various research projects that they're carrying out on this very important topic.
Historically, healthy, fertile soils have been the main reason behind the rise of civilizations. We had the Egyptians, the Mesopotamians, the Indo-Valley, the Incas, and many more. But likewise, soil degradation has also been the trigger for the collapse of societies. We have the example of the Greeks after centuries of soil erosion. Also, more recent disasters. For instance, the Dust Bowl in the 30s, with massive dust storms due to wind erosion of bare soils. This was after just a decade of overfarming, deep ploughing, and loss of organic matter. These poor soil management practices ended up causing enormous economic and agricultural damage. Human well-being relies on healthy, non-degraded soils. To put this in context, soil degradation is globally responsible for the loss of 12 million hectares of productive, fertile agricultural land. This is more than the area of the whole of Ireland being lost every year. So it's important to treat soils as valuable resources and manage them sustainably for future generations. Healthy soils can be defined as multifunctional living systems, with a continued capacity to sustain plant, animal, and human life. Only living things can have health, so it's important to see soils as living and diverse ecosystems, full of life. Soils are multifunctional. They produce 95% of all the food and fibre for humans and livestock, but they also provide other important services for society. They regulate climate by capturing CO2 through plant photosynthesis and keeping that carbon in the ground as soil organic matter. Soils are the place on Earth with the highest biodiversity, and they are the source of many useful chemicals and medicines. There are more living organisms in a handful of healthy topsoil than all the humans who have ever existed on Earth. Soil microorganisms also recycle our waste byproducts from food production into useful nutrients for plants. And healthy soils also purify water and minimize the risk of flooding downstream in our towns and cities. There are many different types of soils, each one with a different ability to carry out these functions. Here in Ireland, our main issues with soil degradation are compaction, mainly from farm machinery, loss of nutrients contaminating our water streams, and erosion. Society needs to better understand the practices that lead to soil degradation, and also those that can heal and improve our soils. That's what we are here for at Teagasc.
Hello, I'm Guy Lagrange. I'm a third-year PhD student here in Johnstown Castle, and I'm working on plant biodiversity in Grassland. I'm going to talk to you today about the productivity function of the soil, illustrated by these animals here that produce milk to feed the humans.
Can you imagine growing vegetables, barley or wheat without soil? It is actually the same for the grass. The grass needs good soil for growing. What is a good soil? A good soil would be a soil providing them in nutrients and also that can hold the water, so with good water retention you can balance the periods without rain or with too much rain.
The best experts we have today to take care of our soil are farmers. Every day they take decisions to keep the soil fertility high. This way they are sure they will have good grass to feed their animals, and so they can sell good milk or good meats for everybody to eat well. But the climate is changing. We expect less rain in summer, more rain in winter, and there are concerns raised about the use of fertilizer that could produce greenhouse gases. To answer these questions, Then we are trying to develop here in Johnstown a grasslands that could cope with that, so cope with less fertilizer and less rain in the summer. We are testing the ability of multi-species swards to cope with droughts and also with reduced fertilizer use. To do so, we mix grass, herbs and legumes like clover, and so interacting together they can benefit to each other and maintain good productivity with low inputs. Clover, for example, can increase soil fertility, and other species like herbs have long roots that can bring carbon deeper into the soil. We will now see why carbon storage is an important thing.
Hi, my name is James Rombo and I am the soil carbon sequestration technician here in Johnstown Castle. The great environmental challenge of this century is the need to stabilise atmospheric concentrations of greenhouse gases. In order to achieve this, we, the caretakers of this planet, need to reduce our dependence on fossil fuels and find ways of removing greenhouse gases from our atmosphere.
The scope of the issue we face is very challenging. Currently, sources of greenhouse gas emissions from human-related activities release 34 gigatons of CO2 into the atmosphere annually. At present, there is no hope for a radical reversal of these rates of emission. Aside from reducing our fossil fuel use, there is one method we can all use to try and stem the rate of greenhouse gas growth. This involves the sequestration or capture of atmospheric CO2 as organic carbon in our soils. Carbon stored in soils is very beneficial. It improves their structure, their water holding capacity, their productivity and their biodiversity. Europe's recent Green Deal aims to prevent climate change by bringing down carbon emissions significantly by 2050. Beef and dairy production are the largest Irish agricultural industries and the main cause of the sector's emissions. Reducing the national herd's carbon emissions as much as possible while feeding a growing population is going to be very challenging. Here at Teagasc Johnstown Castle, we use a range of equipment to monitor and track CO2 and soil carbon, such as eddy covariance towers, elemental analyzers and pneumatic soil corers. These enable researchers to look into improving the national herd's carbon footprint. The main strategies being investigated in order to achieve this include improving efficiencies, adopting low emission technologies and carbon sequestration.
Okay, so I'm going to talk to you briefly about what exactly soil carbon sequestration is and how it happens. So we have our plants, which, as we all know, photosynthesize. They use solar energy combined with carbon dioxide, which is our big problem word at the minute, and water to create simple sugars such as carbohydrates. So they use these simple sugars to grow biomass. This biomass is above and below the soil. Now, following decomposition, it eventually becomes a stable form of carbon in the soil. I just demonstrate here 3 types of sod that I have, one from a woodland, one from a long-term grassland and one from a tillage soil. So we see here with the woodland, which has been woodland for 20 plus years, we have deep roots which go right to the bottom of the sod. You can see it's very stable, it's in one piece. The water is well held within the soil. There is ample pore space for water to move through, but not enough pore space for water to wash away the soil. Here we have our grassland, which again has been grassland for 10 years plus. You can see it's much shallower. There's quite a dense biomass on the top here. They're quite deep rooted, not as deep as the woodland, but it holds firm together. It's well structured. Again, another very good method to sequester carbon. And then we have our tillage. And the tillage, as you can see, it's very crumbly. It doesn't really hold its structure too well. There's not really a great root mass. Very compared to the woodland, there's very little biodiversity in the soil. However, this is the sod that has the most potential to sequester carbon. With careful management, such as minimum tillage, or we're looking into very deep ploughing, etc., there is an awful lot more potential to sequester carbon long term in tillage soils. Okay, so
when we hear carbon dioxide, the majority of us think of it as being a bad gas, but in actual fact, it's a normal constituent of the atmosphere. The issue is when there's too much carbon dioxide. So I have set up a little bit of an experiment here just to demonstrate exactly what happens when there's too much carbon dioxide present in the atmosphere. So we have a little temperature sensor here that's reading 19.2 degrees at the minute. Both these bottles have distilled water and are filled with ambient air. So they have normal levels of carbon dioxide in them. I have two effervescent tablets here that I'm going to drop into one, which will release carbon dioxide. I will close the lid on both and then add a heat source. And normally what we should see is an increase in temperature over the standard in the carbon dioxide bottle versus what is normal ambient air. So I've just dropped the effervescent tablets in. You can see it's starting to fizz now. I'm just going to place the lid on top and let the carbon dioxide build up inside the bottle. Okay, so I'm removing our thermometer, adding the lid to our control, flicking on our heat source and placing it in front. So now in theory our carbon dioxide bottle should heat up faster and to a higher temperature than our control. The lamp here actually represents the sun, which was both a light and heat source, and the two bottles represent our atmosphere under normal conditions and under exaggerated carbon dioxide conditions. Okay, so we've left the experiment overnight. It's actually got a bit cooler in here. The heating's been off, obviously. So this is our control. We are now just about 17.5 degrees Celsius in our control. And if we look at our CO2 bottle, then we are up around 26.7 degrees or so. That just goes to show a temperature difference of 10 degrees Celsius, and that's just here in the lab in front of us. That's what's happening in our atmosphere at the minute when we are emitting such amounts of carbon dioxide into the atmosphere.
Hi, my name is Ian Kavanagh and I'm going to be talking to you today about the importance of soil and its role in nutrient recycling. Irish agriculture, be it animal or crop production, produces a lot of organic residues in the form of animal slurry or incorporated into bedding to create farmyard manure. However, other byproducts such as food processing, waste management and industrial residues also have to be dealt with. The incorporation of these organic residues into the soil is one such method to reduce the organic residues from animal farming and food production. These organic residues are a good source of organic matter, carbon and nutrients in the form of nitrogen, phosphorus and potassium. These feed soil biodiversity and help sustain healthy crop production. This is of vital importance as this nutrient recycling helps close the carbon, nitrogen and phosphorus loop and promote the farm-to-fork cycle, while also helping to reduce the use of artificial chemical fertilisers. With this in mind, Teagasc is running a number of experiments and field trials to assess the impact of these residues and the best way to utilise the nutrients available in the soil. Trials such as the EU-funded project Nitri2 Cycle is investigating new bio-based fertilisers for their efficacy in grassland and arable crops. They also examine their benefits to soil health, such as increasing soil organic carbon. Other field experiments have also looked at acidified and amended cattle slurry and its utilisation and incorporation into the soil. However, none of these residues would be utilisable if it wasn't for the wide variety of microorganisms that call the soil home.
Hi, my name is Aoife Duff. I'm a soil microbiologist here in Teagasc, and today I'd like to talk to you about soil biodiversity and how it contributes to soil health. As you've already heard, carbon sequestration, nutrient cycling and all of the processes mentioned so far are very important for soil health. But what makes these processes happen in the soil? Well, some of the processes take place via chemical reactions, but the majority of the processes are driven by what we call microorganisms. In fact, in 1 teaspoon of soil, you can find millions of species of microbes, and together it contains billions of microorganisms. So essentially, the soil is teeming with life. So here in Teagasc, we are doing lots of projects to work on soil biodiversity. Paula Roja is our technician. What are you working on, Paula?
I'm working in how the soil microbial community can divert the agricultural greenhouse gas emissions coming from the soil.
You may ask me, how can you tell how many microorganisms are in the soil? Well, not too long ago, the only way of identifying a species and discovering what they did was to grow them on lab media. Media is a combination of different elements like carbon and nitrogen, which the microbes require in order to grow. The problem with this method was all microbes may not eat the exact same food or elements, and so the scientists discovered they were only growing about 1% of the microbes present in the soil. Microbiologists started to find a new way of looking at microbes in the soil.
Hi, my name is Circa Kelly. I'm a PhD student here in Teagasc, Johnstown Castle. My project looks at how soil microorganisms make nutrients available from soil organic matter in order to help plants to grow. Today, I'm going to show you how we extract microbial DNA from soil.
Circa Kelly is demonstrating a DNA extraction. Here she uses a fast prep machine to vortex or shake the beads in the tube and chemically and mechanically burst open the cells to release their genetic code.
Hi, my name is Kerry Ryan. I am a PhD student and I study microbes in the soil. I'm particularly interested in how mixed species grasslands affect what kind of microbes we find in the soil, how they work, and how they are affected by drought. Today, we're going to look at how DNA can be separated through gel electrophoresis.
Kerry Ryan then carries out a gel electrophoresis so that we can visualize the DNA. Finally, with a combination of different techniques and sequencing technology, we were able to read their genetic code, which told us all of the genetic information of each species of microbe. So this is just a taster of the world of microbiology and soil biodiversity in Teagasc. We hope that our projects will help farmers employ the best management techniques possible to keep the soil biodiversity balanced. This in turn will keep the soils healthy in order to carry out vital functions that will help the farmer and the environment.
Hi, my name is Damien and I'm a soil scientist in the Agriculture Catchments program. So as you have heard by now, soil has many different functions. One of them is the provision and supply of clean water. Only 3% of Earth's water is fresh water, and we can only access and use a tiny fraction of that fresh water. Imagine that this tank contains all the water on Earth, and this little bottle would be all the fresh water on Earth. And then this tiny little teaspoon is all the water, fresh water we can access for drinking and other purposes. The amount of this water is shrinking every day as a consequence of things like climate change and water pollution. Fresh water supply is continually collected, purified and distributed in the natural water cycle with the help of soils. Soils are Earth's largest natural filter, and capturing water is one of the most important roles that soils play in our ecosystem. Soils capture water through their pores. Pores can be any size from small, microscopic holes to large worm-like holes and moist tunnels. The size of soil pores is determined by soil texture, which is a ratio of different soil particles such as sand, silt and clay. These particles stick together with the help of biological life and organic matter, which provide the glues to form tiny crumbs called aggregates. Aggregates are the building blocks of every soil, and they contain lots of pores and cracks. Aggregates form soil structure, which increases the ability of soil to capture and hold water. However, the ability of soil to capture and purify water is limited, as I will show you now by pouring this dirty water onto 3 different types of soil. Sandy soils have many large pores. While they're good at capturing water, water moves quickly through them. This does not allow soil enough time to fully purify on its way downwards to groundwater. Loamy soils have both large and small pores. While water moves through them quickly, some of that water is also filtered. Clayey soils are sticky and have lots of small pores. They are very good at purifying and holding water, but they are slow to capture water. Water that is not captured by soil runs off the soil surface until it reaches a nearby source of surface water, such as stream, river, or lake. This water can also cause floods during heavy rainfall events. The runoff water also picks up soil particles with all the nutrients and chemicals attached to them, transporting and depositing them into receiving waters. This has a very harmful effect on aquatic plants and animals. Healthy soils are full of life and have good structure and organic matter content. Because of that, they have lots of big and small pores. Therefore, it is important to minimize the disturbance of soils. Frequent tillage or compaction by heavy machinery destroys soil structure. These soils will only have small pores. Land surface cover is also important. Healthy soils covered by plants are most efficient at capturing water. Plant roots keep the soil together, dead roots provide soil organic matter, and together they make the soil resistant to erosion and better at capturing water. The above-ground biomass, like trees and plants, reduces the devastating impact of rain and slows down the flow of runoff water. It is also important not to fertilize soils more than is needed by plants, especially with nitrogen and phosphorus fertilizers, or to spray them too much with chemicals such as pesticides and herbicides. This is because excess fertilizer and chemicals can enter and contaminate the groundwater or can be washed off the surface, polluting our streams, rivers, lakes, and seas. Remember this teaspoon of water. So how do we prevent it from shrinking in future? One of the answers is to keep our soils healthy.
Hello, my name is Lillian O'Sullivan. I'm a research officer here in Teagasc, where my work focuses on sustainable soils and land use. So now you have seen an overview of the different soil functions. We know that soil functions are critical to sustain life on Earth, and my work focuses more specifically on how these soil functions work together at larger scales. It is important that we manage the system sustainably to maintain soil health, to guarantee the delivery of these critical soil functions into the future. So how can we support this through our research? Process knowledge can explain how each soil function works, and each soil function is a very complex process in its own right. For example, the process of cleaning and purifying water as it moves through the soil matrix, as Augie showed you earlier, or how the soil accumulates and stores organic matter, which is really important for climate regulation, as shown by James. Understanding these processes helps us to explain how much a particular soil in its environment interacting with management can deliver a soil function. Recently, as part of an EU-funded project, Landmark, we developed the Soil Navigator, which is a decision support tool that can help farmers and advisors optimize their soil functions together. So essentially, you input some data and you ask it to assess your soil functions, and then it allows you to optimize based upon your requirements. So if I have an issue with water quality, I will ask the Soil Navigator to optimize in favor of that soil function. One resource I often use is our Irish Soil Information System. It's a fully interactive map. Anybody can go online and learn more about the extent and distribution of the different soil types that we have or in your own area. Here in Johnstown Castle, we aim to generate science that can support sustainable land management. And remember, caring for soils is caring for life.